Hello everyone, my name is Gabriel Moraes and I'm a pharmacy student advised by Professor Marcelo Távora Mira, PhD. This study was entitled Association Analysis between AGO, EGR2 and CDH18 exonic variants in leprosy, funded by Fundação Araucária. The background: leprosy, also known as Hansen's disease, is a chronic infectious disease caused by Mycobacterium leprae, an acid-fast bacillus which infects mainly Schwann cells and macrophages. Leprosy was the first human disease to which an infectious agent has been described. It happened in 1873 by Gerard Hansen. Mycobacterium leprae has a quite long incubation time and it cannot be cultivated in artificial media. Its clonal structure worldwide, added to observational studies and new molecular methods, simulate the understanding of how the host genetics has been associated as an important factor in leprosy development. The objective of this study lies at the comprehension of the genetic control in the host susceptibility to leprosy, applying association analysis between the disease and the variants positioned in translated and untranslated regions of the ADO, EGR2, and CDH18 genes. This is a family-based association study, and 177 peripheral blood samples were recruited from families living in Vila Santo Antônio do Prata, a former leprosy colony that remains isolated and hyperendemic for the disease today. DNA extraction was performed, followed by DNA quantification and next-generation sequencing. The variants data was first aligned, filtered, and then the variant analysis was performed. The GC content was around 44%, which was expected, and most of the reads have an average quality greater than Q30 in the Phred scale, which means that the reads have an accuracy greater than 99.9%. 28 variants with minor allele frequency greater than 0.04 were identified in at least 8 informative families, and 103 variants with minor allele frequency less than 0.03 were also identified in the sequenced genes. The transmission disequilibrium test revealed one variant associated with leprosy per se. This variant is located in the UTR region of the AGO gene, and it indicates a possible harmful impact in silico analysis, and it also influences gene expressions, which means it presents a potential functional impact. Linkage disequilibrium analysis was also performed between the variants of this study and a variant association in previous studies. It resulted in a weak association in two Asian populations. As conclusion, this study identified a variant with potential impact in the ADO gene expression as predicted in silico, and validated the association between the host genetic susceptibility to leprosy and the ADO gene. The results obtained from this study could be further validated in populations with distinct genetic background. That concludes my presentations. I sincerely appreciate your interest and attention.